Alright everyone, so what we're going to be doing in this series of lecture videos is I'm going to be introducing to you some basic concepts related to genetics and the study of how information is passed from one generation to the next. So in this series of videos, what I'm going to start out with is talking about the work of Gregor Mendel to give us a historical context of how we came to understand what we see today. Then I'm going to do a video that talks about the probability of these different scenarios and introduce a Punnett square. Then we're going to look at exploring Mendelian genetics. So we're going to be looking at some kind of interesting applications for Mendelian genetics and some variations in how genes may be expressed. And then lastly, we're going to talk about meiosis, and we're going to go into a little bit more detail about that process, given the context that we now have about genetics. So let's begin by talking about what genetics is. Okay? So to really get to genetics, we need to ask a question. And the question that I'm asking is, what is an inheritance? So we can define an inheritance from other contexts, things that we know. And an inheritance is just anything that's passed down from one generation to the next. So some things that you might say are inherited, money, right? So if, for example, you have like your fourth uncle twice removed that had nobody relative to you and they left for you, $1 million, that would be an example of inheritance. You can inherit property. You can inherit knowledge, for example, like I'm giving you access to a lot of material and information that we've learned already, so the next generation is responsible for continuing to learn. And we're talking about a genetic context and that we're talking about biological systems, we're talking about the inheritance of traits or physical characteristics. So when I had talked about physical traits, right, we're talking about the biological inheritance, like what you as an individual, for example, has received from your mother and your father. So what we're going to be looking at with inheritance is we're going to first kind of take that physical trait, keep that in mind, like that's the thing we're looking at, that phenotype, that physical characteristic, and we're going to dive into some of the histories and nuances that we've gleaned from work done by this old school dude. All right. So we know from looking at meiosis that the experiment, or that every living individual has a set of characteristics that they inherit from their parent or parents, right? And so I say parent because you could be an individual that has been reproduced asexually, so you have the same exact genes as your parent does. Or if you have multiple parents, so a mother and a father, for example, you have a mixture of the different genes, and that you are expressing some of the alleles that are genes that came from your mom and some of that material that came from your dad, right? And that's why when you look at yourself and compare it to your parents, you look kind of like both, right? You have traits that you express from both of them. So when we're talking about the process of actually carrying this information from generation to generation or from parent to offspring, we're thinking about this concept of heredity. So the idea that you inherit something or the heredity of the situation, right? That whatever gene you're getting is going to lead to an individual's combination of alleles, so a combination of copies of traits. And then lastly, just the study of genetics, right? When we think about genetics, think about it as the study of inheritance, right? The idea of understanding how information gets passed from one generation to the next and what are the factors that can influence how that information gets passed. So the study of genetics is relatively young in terms of our study of science, but it's kind of this core revelation in understanding how organisms change from generation to generation that's really redefined the genre of biology as a whole. So we're going to focus on initially looking at the experiments done by this individual, Gregor Mendel. I'm sure some of you have heard of him. He's pretty famous. We call him essentially the father of genetics because of his meticulous work that led to a broader understanding of how organisms are carrying information from generation to generation. He laid the foundation for what work that other individuals did, and it was part of his work that then came together with a lot of different other researchers in different branches of biology that led to our greater understanding of inheritance as a whole. So just a little bit about Gregor Mendel. He was born in the 1820s, 1822, and he lived most of his life as a monk. And he was a monk that was in now what we would call the Czech Republic. So like most religious individuals at this time, they were expected to be kind of this sect of individuals that were well educated, that had a lot of time to devote to studying the history, math, science of the world at the time. So Mendel, like most, was a brilliant individual. He left his hometown, went to the University of Vienna, and there he worked as a teacher as well as a researcher studying specifically math and science. He had a great proclivity for those two areas of study. He also had other interests. He was an avid gardener, and he took care of his favorite foods, which was the pea. And he also ended up taking care of the gardens at the monastery that he worked at when he left the university. This is where he did most of his work, was in a monastery in the Czech Republic. And so what did Mendel do? So Mendel was, like any good scientist, very particular about observation. He would spend a lot of time observing the organisms that he was working with, especially the plants that he was using to grow for food. And he noticed there are some differences from plant to plant. There were some things that would change. When he would plant them, he would see some things show up. He would see some stuff disappear from generation to generation. And so it was those initial observations that led him to start to set up a more structured way of eliminating some variables to try to do a study on these different organisms from year to year. And so what he ended up doing was taking the pea plant. So he used the pea plant because it was, one, relatively fast to grow, two, it produced fruit, so he could study that. It had a lot of different it had a lot of different traits from plant to plant that were really useful and helpful for do this. So things like studying stamens, studying those kinds of figures were interesting to him. But eventually what he noticed is that if he kept the same individuals and only allowed them to breed with themselves, so that is, in plants they're able to use their own pollen to fertilize themselves and create offspring that are essentially genetic clones of the parent, that he could get generations of plants that only exhibited particular traits and that they would stay true to that. And so what he did is what we call true breeding. So what he did is he allowed the plants to self-pollinate so that they would always exhibit the same traits. And then so when the fertilization event happened, the male and female reproductive cells came from the same individual, which plants can totally do that. They're totally okay with it. And so that kept the trait relatively consistent, and he would eventually see lines of plants that only exhibited one example of a different trait. I'll get to that in a little bit. So because peas can self-pollinate, right, meaning that they can take their own information, he had to essentially fix the plants in a way. He had to go in and remove the anthers from some of the pea plants to make sure that they didn't self-pollinate, and so that he could control which flowers were mated with which other flowers, so he could control which plants were mated with which other plants. And so because Mendel did this meticulous work to make sure that he was crossing plants that had particular traits, it led him to really look at different characteristics that would show up from generation to generation. And so a trait, remember, is a specific characteristic of an organism that varies from individual to individual. Okay, so if you think about a trait as a category, maybe that'll help. So for example, I can think of trait as like a car, right? A car is something that you can drive, it's something that you carry you from point A to point B, right? But if you look at all the different cars on the road, they all take a bunch of different forms. They're still all cars, but they do slightly different things, right? An SUV does something different than a pickup truck, that is something that is different than like someone driving a sports BMW or something like that, is that they're still all cars, but the way that they're expressed are all different from each other. So he would see the same thing in plants. So they all had like the same necessity function, right, to say do something for the plant, but they took on a different form. So what Mendel did is that he ended up growing seeds from individuals that exhibited what he said were true traits, right? That they didn't have any variation from generation to generation, and that all the offspring looked just like the parent. And so what he did is he ended up looking at 7 different traits. So he kind of whittled down all the potential traits he could look at, and he was just focusing on these 7. So the plants themselves that he ended up producing from the parents, so that initial generation, was a hybrid. He would take traits from one individual and traits from the other and cross them together to see what kind of offspring was produced. And so with each generation, he would produce what we call an F generation, or a filial generation. And so the first one is the first filial, the second one is the F2 or second filial, and we continue that trend if we say go to our F67th generation, which would still be our 67th filial generation. And in each generation, he observed the characteristics of the plants and kept track again of what ended up reproducing with what. So in Mendel's work, these are the 7 traits that he looked at. So I don't expect you to memorize them, but you should be aware that he had 7 different categories and 2 different expressions. So for example, height, he had like a tall one and a short one. So he had plants that would grow a few feet tall, and he has some that would be like short, kind of shrubby, little bushy ones. Okay. He looked at seed shape, seed color, seed coat, pod shape, so whether it looked like a sugar snap pea or it was like a kind of wrinkly skin, if it had a green or yellow pod, or flower position, so if it grew at the top of the plant or if it grew at the side. And so what he did then is he took each of the groups and crossed them together. So for example, he took a tall plant and crossed it with a short plant. So taking that parent generation and producing the F1 generation. And so when he ended up taking these different traits and putting together the two opposite traits, he noticed that all of the offspring only exhibited one of the two traits that he knew were present. So for example, all of the offspring were tall, instead of some of them being tall, some of them being short, or what he had probably expected for the offspring to be somewhere in the middle, to be an intermediate. So this was really shocking for him. He was like, whoa, like, what, man? Like, I don't get it. It was where it wasn't meeting his expectations, right? It's challenging his hypothesis and what he would expect when, you know, he'd already seen this kind of blending of characteristics that would happen in animals that didn't show up in these plant populations. So he was kind of stumped. So initially he just thought like, okay, well, they disappeared. And so he ended up concluding from this initial step a few different things. So one of those is that there is something that is passed from generation to generation. He wasn't sure if it was, he didn't know anything about DNA at the time. He didn't know anything about genes. He didn't know anything about meiosis. That was not discovered and explained yet. But he did know that there was some kind of factor that was getting passed down from generation to generation. There was something that was getting carried. And so this factor we now know as biological inheritance, and we know that factor is a gene. Remember, a coding portion of a DNA sequence that codes for a particular protein that leads to a particular phenotypic expression. So he knew that those were present, right? That there was something that was getting passed from generation to generation. And so we know that those are the genes, and that there is a gene then that's coding for height, right? And that one of the ways that gene is being expressed as height is as a tall plant, and one of the ways it's being expressed is as a short plant. Okay, and we call those different ways of expressing the gene an allele. So again, like in our car, Ford truck would be an allele of a car, right? It's one particular type that codes for the same thing that is a vehicle that gets you from point A to point B, right? Another way that allele can be expressed is as like a, I don't know, like a little Fiat, right? Still able to get you from point A to point B, but functions very different than a truck. Okay, so they're still the same trait, right? They're still a vehicle, or they're still height in the plants, but you just have tall and short, and that for some reason, we're seeing the expressions show up this way. The other thing that we noticed, or was able to determine it, there was some principle of dominance. There was something that was telling Gregor Mendel that this particular trait was more expressed than some other trait. And so I'll talk more about that one and kind of how we can see that dominance and expression play out, but we're going to look at an example of the different allele types. So just know that if you've got a dominant allele, right, if an individual has for that particular gene the one that say is dominant or the one that's going to be readily expressed, right, that dominant allele will always show, right? If you have a dominant allele, you will always express that dominant allele for the most part. Again, there are exceptions to all of this, but generally if you have a dominant allele, you will express it. If you have a recessive allele, that means you will not express it. It will not be seen as a physical trait unless it's the only kind that you have. So if you have one dominant and one recessive, you're going to express the dominant. But if you have only recessive alleles, you're only going to express the recessive characteristic. All right, so here's the example I want to talk about just kind of with expression. So if I have my two alleles, right, one that's dominant and one that's recessive, let's say in these bunnies, they're freaking adorable, that I have a brown allele, right, and I have the white allele, both for fur color. And that if we say the brown allele is dominant and the white allele is recessive, then we know that in the offspring, if you are brown, you're going to have at least one of those brown alleles. And if you have white, you're going to then have only the recessive allele. So if I cross these offspring, right, I would assume that I would mostly get what type of bunny in the end? I would most likely get brown bunnies, right, because they are going to have that dominant allele. And one of these is going to get passed on to the, or they're going to get these passed on to the next generation. And we'll look at that in more detail later. But just know that if you have the dominant allele, that's the one that you're going to express. So with Mendel's work, there are some other things that he did, is that after doing that first generation, that F1 generation, he just wasn't satisfied. So what he did is he ended up taking those offspring from the F1 and ended up making, crossing them with each other. So he put these F1 plants together, and he ended up noticing that, yes, most of them were tall, but all of a sudden, what? He would get short. There would be a short plant that would show up in this mix, okay? the recessive trait, or whatever he thought was recessive, or whatever disappeared, showed up again. And that was, again, super shocking. Like, it was gone, and now it suddenly happens. Okay? Most of them are tall, but now this little one still shows up again. So what's going on there? And what he ended up concluding is that recessive alleles don't disappear, that factor didn't go away, and that there was some kind of dominant trait that ended up covering up or ended up being expressed over the recessive one. So how do these recessive traits end up showing up? And it's basically due to the process of meiosis and what happens with independent assortments. The idea that depending upon how things line up, what the offsprings, what the gametes are going to have. So what he ended up concluding is that F1 generation was a hybrid, that there was an allele that was tall with an allele that was short in each of the individuals, and that when they formed their gametes and they came together, they ended up producing mostly individuals that had this dominant allele, right? But one that didn't have it, so it expressed the recessive trait. And so Mendel's work helped us demonstrate that some kind of process was happening that caused these things, these factors, to separate, and then when they came back together again, they led to variation in expression. So we had what we call segregation, which is, again, the separation of alleles during gamete formation. We talked about that with meiosis and independent assortment, right? And that during anaphase 1 and anaphase 2 that we're splitting first that tetrad, and then we're splitting sister chromatids to end up with individuals that have that reduced number of alleles in the final offspring. So the F1 plants could make two potential factors, right? They could either carry the tall or the short to the offspring, but when they come back together, we've got that tall trait again. So this leads to a little bit more terminology. So what we noticed is that when we crossed the F1 and we had our two offspring, segregation occurred, right? These gametes would only get one of the alleles, and depending upon how they get put together, you're left with a mix of individuals that would express that tall trait, and then some that would express that short trait. So we call individuals that get two of these dominant alleles homozygous. If they get one of each, we call them heterozygous. So if you get two dominant alleles, we call it homozygous dominant, and two recessive alleles is homozygous recessive. And we'll talk about that in a little bit more detail later. So again, with segregation, as we're going through this process, this is meiosis, right? That's what's happening here. And then this is fertilization as our offspring come together. Okay? So that's a little bit long, right? But hopefully now you have a greater understanding of kind of where we're starting looking at genetic variation. And so what we're going to look at next is we're going to be looking at the probabilities that are aligned to this segregation process. What's the likelihood that I would end up with offspring that are going to all be short, given that I have most of my gametes having this tall allele, and that the chances of this happening are probably relatively low? So what's the mathematical model that's behind that? So when we're talking about our final traits later, right, we're still going to continue to refer back to the pea plant as our model because it's the best model. Okay. So as we go forward, right, if you've got questions or things, please let me know. I'm happy to answer them. Right, so good luck.